Hi guys, we're back. This is going to be a short video on the making of gametes, sperm and eggs. Gametogenesis is just the conversion. This is all basic bio. If you feel confident with it, you don't necessarily have to spend a lot of time studying it. But it's the conversion of your diploid germ cells. You have these immature germ cells, and they turn into mature ova and spermatozoa. So that's gametogenesis. It's not a gamete until it can combine with someone else's gamete and make a person. This occurs via meiosis and only in germ cells. The result should be, we hope, if it's a euploid sex cell, 23 chromosomes. Two helpful concepts. I'm just throwing them out there. Don't get too wrapped up in them. But every time a haploid cell is made, the DNA deck is shuffled. That's called crossing over. And it means you don't ever get two gametes that are exactly alike. They're all unique little snowflakes. When you get identical twins, it's because a single gamete or fertilized ovum split into two identical cells. But each gamete should be very unique. However, even though we're shuffling that deck so that mom's genetic material is randomized a little bit, there are genes that are close together on chromosomes and they tend to stay together, and that's called gene linkage. For the purposes of this course, you do not need to get too involved with that. If you move forward into genomics, or if you're weird like me and you just take an interest in the science of it, you might find literature on linked genes in different health conditions. It's interesting, but anyway. Spermatogenesis. Spermatogonia are immature male germ cells. They are produced when your baby boy is a fetus, and they remain dormant. They stay these little immature germ cells not doing anything until puberty. Then they go through a rapid period of change. There's a lot of mitotic division, where you make more diploid cells with 46 chromosomes just to increase the number so that you have more cells going through meiosis. It takes a lot of sperm to match that one egg. So men have to make a lot of them. Meiosis in 1 and 2 are a continuous process. They can take anywhere from days to weeks. That starts in puberty. It goes until the end of a male life, theoretically, although sperm quality and quantity do diminish with age. It takes about 2 months for each one of these cells to mature. And it's all regulated by those director genes telling them when to stop and when to go. But for the male, it's mostly go, 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 go. They need to make a lot of sperm, otherwise the species dies out. Oogenesis, on the other hand, is a little trickier. You start with an oogonium, an immature female diploid germ cell. These are all made in fetal life, and they start meiosis 1. They go through that first checkpoint during the end of fetal development, and they get arrested in prophase of meiosis 1 for years. If a woman is 52 years old and she's hasn't gone through menopause yet, and she still ovulates, maybe more infrequently, she's dealing with 52-year-old or 53-year-old eggs. Really, they started forming in fetal life. So there's a lot of room for errors to occur at that point when it has to go through the rest of cell division. Breakages, as that egg lays dormant there. There can be rearrangements of chromosomes, and that's where you get that increased incidence of trisomies, monosomies, deletions, insertions, other chromosomal defects. When ovulatory cycles begin, and that happens after puberty, after menarche, one primary oocyte, will divide into a secondary oocyte in a polar body. So that's kind of what you see here. Okay, you have one cell and then a polar body. Then that divides again, and you have an egg cell which will become a mature egg cell, and there are these 3 polar bodies. So where a man makes sperm, each diploid cell ends up becoming 4 spermatozoa, and they will all mature. A woman will make 1 mature egg cell from each oogonium eventually. The other 3 cells involved in that division become polar bodies, okay? And they do not become eggs that can be fertilized. So the big take home from oogenesis is that because the oocytes are arrested, they have already developed, there's just all that time for chromosomal breakdown and rearrangement of chromosomal material in ways that you don't necessarily want it. And that is the end of gametogenesis. It's really the end of the first half of the content. We'll pick up again with epigenomics, and I'll see you in a bit. Bye.